All right, hello. I'm going to try to help you out with the sickle cell disease lab that you're supposed to do today. I did attach some answers to the normal hemoglobin. I'm just going to walk you through some of the procedures for this as well. So in procedure number 2, it says in table 1, in the row marked mRNA code, write in the correct letters that will match with the nitrogen-based letters of the DNA. Remember, in the DNA, apple always went in tree, car went in garage. Remember, they always matched up with the same letter. Apple and tree, car and garage. They stood for names, adenine, thymine, cytosine, and guanine, but just we always remembered that mnemonic device, apple, tree, car, garage. Well, we are now making the mRNA copy, which remember is half the ladder. Remember the half the ladder they cut in half, that Three Stooges video I showed you? Now, mRNA, to make a copy, it has one difference, and they told us this in the video, and I explained it, that instead of apple and tree, it's apple and under. A matches with U. If there's a T in DNA, it matches with an A for RNA, C with G, and G with C. So it's going to look, so here's the DNA code. So it's going to look like this. G is garage, is going to give us car. G is going to give us C. C, G, A, G, A. Now that mRNA is then going to leave the nucleus and go to the ribosome. Now in the ribosome, tRNA is going to decipher what the code was. tRNA it's an RNA, so it has that U, so U with A again, and G with C. So if we're going to do the tRNA, it would look, let's change my color here, it would look like this. G, C would be with G, 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 C, U, U, C, U, 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 U. That's the tRNA code. Next, the next instruction it tells us to do is, so now we got the tRNA. Now, every 3 tRNAs have a protein part attached to them. Well, tRNA codes from the mRNA, actually. So if we look at that mRNA, I'm going to circle every 3 one of them. Because every 3 mRNA, each of those sets gives us something called a codon, which is what the tRNA codes to make the protein part. So there's a codon, there's a codon, there's a codon, and there's a codon. Sets of 3. 3, 3, 3, 3. There's 4 codons that tRNA is going to code from the mRNA to give us the protein part. Now it tells us in the data table what they just made up what these protein parts are. But the protein parts, protein part A, B, C, and X, that's the sickle one. If the mRNA is CCC, that is protein part A. Well, that's actually what the first one is, CCC. So CCC would give us protein part A. GAA, which happens twice in a row. If we look at this again, GAA gives us protein part B. So let's go back to that. GAA gives us protein part B. GAA again, protein part B again. And finally, AAA, we go back and look, AAA gives us protein part C. So let's write that, protein part C. Eventually, those proteins are going to make the hemoglobin, normal hemoglobin, red, normal hemoglobin is round. Has a little indentation kind of in the middle. Round, a little indentation in the middle. So maybe looking at it from the side, round. So round, normal sickle hemoglobin DNA. If you compare the DNA, and the reason these are all different, this DNA is different than sickle hemoglobin, is if you look at it, it's because the 2 letters, 2 letters are different. T's got mutated to A's. And just because of that, those 2 being mutated, it's going to give us a different protein part, and it's going to cause sickle cell anemia. Not a good disease. And we're going to play some games and do some title quests also on sickle cell anemia. Hopefully that helped you. You're going to have to do the sickle cell side on your own. Good luck. And as always, email me if you ever have any problems.